Thank you so much, everyone, for coming. We're, really delighted to have Samaya here today. We met earlier this year, and I was just really excited about her work and the opportunity for her to come and share it with you. So, she's a senior group leader, just so I get all the details right into my cheat sheet. She's in a program at the Broad called Ladders to Cures. It's a specific accelerator that works across both academia and industry, as far as I'm aware. She's also an associate member of the Cancer Data Sciences program at Harvard. And she leads her own team, which utilizes her computer science background and really applies it to understand rare diseases, especially rare cancer, which is really, I think, very important and exciting area. She has a very strong background in ML, AI methods development as well, and has developed a platform that really helps connect genomic level data to actually protein level data. I think that's really something that's a major gap in the field that her work has addressed, and she'll be able to tell us about. She's earned multiple awards, including the Broad Institute Spark Award, the Merck Institute Award for Transformative Technologies in Healthcare, the Broad Knight Award, and also is a recipient of the Broad Institute Staff Scientist Distinction Award, in specifically for collaboration.
Yeah,
which I thought was
a lot of collaborations. Yeah. A lot of collaboration.
Important part of what we all do as scientists, and also at Brown is really important to our community. So with that, I'm very excited to hear.
Thank you, Erica. Thank you.
Her work in her group.
How can I confirm that this is working? Oh, I really hope, and I really wish nobody would be seeing this, but
yeah, we can just that's okay.
Okay, I think I cannot do anything more about it, so I'll just go ahead and start. So thank you everyone for being here. Good afternoon. And thank you, Erica, for being such a kind host. It's been a terrific day here with students. I loved that part. That was my best part of this whole day. Let's see. This is like very quickly my disclosure slide, and then this is probably the one of the two most important slides that I have in this huge deck. I'll swift through, and that is my people in my lab, and I want to acknowledge their work. Literally everything that I will demonstrate today in certain capacity has have been contributed by them. So kudos to them for their hard work. Okay, so now I know that I have a pretty vague title of my talk, like connecting the dots. So I'll just try to give you a very brief glimpse of what do I mean by it. So currently I am leading the bioinformatics and machine learning team at the Ladders to Cures Accelerator, where our focus is basically nodal biology of rare disease. So what do we mean by that? And that is accelerating the discovery of data, tools, or technology that will help us to converge on a single druggable node for many rare diseases together. Because as we know, the drug discovery is almost impossible and costly and time consuming. So we want to target many diseases at the same time. But I am not a biologist in any possible sense. That is like one piece of information that you must know about me. So I am by training a computer scientist, and after I graduated out of my bachelor's and then my master's, all from Bangladesh, I decided to humiliate myself for multiple times through 10 years of span, where I did my PhD in protein structural biology, my postdoc in genetics, and then I also worked as a professional scientist at the Center for Development of Therapeutics. And now if you take all these words that are bold-faced and underlined in this slide deck and put a connection through them, that's basically what we do. That is the title is about. That is, we connect data across proteins, genetics, therapeutics, and diseases to go to the mechanism using computer science and machine learning and data. So if you go ever to my lab website, it's going to tell that we connect genomics to proteins, and towards that, we do research development. We do develop methods to understand molecular effects of protein mutations, and we also develop machine learning tools for drug discovery. Unfortunately, I don't see part of my slides because of this thing, which I cannot do anything about. Okay, so sorry. Yeah, I'm trying. I was trying to look at you because I want to talk to people, not to the slides, but okay, we'll see. What I'm going to do to begin my talk is I'm going to give you a glimpse of what's going on in each of these different arms within the lab. And then we'll tell you that what's the agenda today. So what we will cover and what we will not cover. So under the resource development, my lab is responsible for Broad Institute Genomics to Proteins platform. And this is what we'll talk about. And then next, under the method development for molecular effect of genetic variants, we have a few things going on. In one hand, we work in developing translatable scores and methods to understand molecular effect of genetic variants. And on the other hand, we do develop methods to translate synthetic variants, which are basically the CRISPR-Cas9 base editing tiling mutagenesis data. And these are the two things I'll talk about today. And what we will not talk about is the molecular dynamics-based understanding of conformational effect of protein that we regularly do. And we also use large language models. I will rarely use AI in this entire talk. I have very strong opinion about using that word, but we'll get to there when I will have one slide with AI in it, or maybe a few more, not one. Okay, so then we do develop machine learning tools for drug discovery. And when I say drug discovery, I mean early stage drug discovery, which is nothing that happens in drug development. Just for your information, what industry does is phenomenal. They do drug development, and we do not do drug development. What we do is early stage drug discovery. That means target discovery and hit discovery that they then make as a drug, which is a whole different thing. Okay, so in that space, we do target discovery, and we'll talk about this very briefly today. We do high throughput experimental technology experiments, which is called DNA encoded library experiment, which generates billions of data point given our drug target and with a small molecule in hand, and we develop machine learning tools for this. And next is something that I'm very excited about, but it's also a very new area, that is we do develop methods to explain what's going inside large language models. So basically, we take out the embeddings and we try to correlate what information is encoded within large language models with the metadata and biological data that we know. So that I call it as explainable AI methods. So, okay, let's go to the first method that we'll talk about. And feel free to ask questions any point. I can see all of you, so I'll answer your question as I go. So Genomics to Proteins portal is a resource and discovery tool for linking genetic screening outputs to proteins. Okay, so now first question that we have to ask, like how to start a research, that was the greatest question that I had today from the students here. That is, what was the necessity of it, right? So rationalize a research project. That is, in the cell, our DNA, proteins, and in between RNA, they don't work in an isolated fashion as we want to do, right? Sitting in our desk and work and do nothing else. But they actually very much talk to each other, and we need to talk to each other too. So DNA and RNA and the messenger, so the instruction manual and the messenger and the performing worker, the proteins, they work in collaborations within the cell. So the idea was then we need to flow the information through these different omics to get to do the overall understanding of how biological process works. And this is the same when we do have an error in the manual, right? So when we have a variation, so we want to translate that discovery into therapeutic hypotheses, we need to do the same thing. We need to take that information through DNA to RNA to the protein. So that is important specifically for two different reasons. First is understanding the structure function effect of protein mutation. So we operate in a rare disease center, and I have the privilege every day, almost sometimes in certain weeks, to talk to patient advocacy group where I'm talking to mothers whose kids are severely ill. And this is what I often observe. So in the genetic variant space, we have nonsense variants, frameshift variants that leads to complete truncation of the protein or aberrant protein. But we have these things called point mutation that changes only one single amino acids, right? And oftentimes, this is what happens, that one single variant has a profoundly big impact on your protein function. And sometimes every individual human being that is coming to us with a certain disease phenotype, let's say 5 people, right? They all have this amino acid change, substitution, or point mutation, but different point mutation, not exactly the same. Now, but exactly the same disease phenotype. And then when we take those mutation and when we map those on the structures, and this is what we observe, that like skipping all the other parts of the protein, they all cluster in one certain region of the protein. And we hypothesize it's because that region of the protein is particularly associated with the function that goes through the DICE function because of this mutation. And that is basically causing the pathogenicity, right? So this is what we observed for this particular protein when we took DDX6X and then we DDX6, which is an RNA helicase, and all the mutations are mapped onto this REC-A domain, which is important for binding with RNA, and RNA DICE function is exactly the pathomechanism behind this disease phenotype that these people manifest. I was part of this study. This was done actually in Arizona in a translational research institute, fabulous work. Now, there is the second reason why we need to connect genomics to proteins, and that is drug discovery. So multiple times, it has been shown that the probability of a target indication pair, which is like target disease pair, moving through the drug discovery pipeline is twofold higher when it is supported by genetic evidence. It has been shown by GSK in 2015. It has been shown by AbbVie in 2019. It has been shown by Deerfield very recently, Deerfield/Broad at 2024. So we basically need this, but I do not know how many of you have ever tried to investigate your target protein by mapping all the mutational data and maybe also functional annotation, all from genetic coordinates to all the way to protein, and you found that it's really not easy to do it. And think about a life without AlphaFold, by the way, in experimental structure, right? It's a real life, really. I lived it, and many here lived it. So that is exactly why we developed Genomics to Proteins portal. Which it does is it takes information from the genomic coordinate, maps it to the right RNA, right transcript, right, which is extremely essential, by the way. Many times patients coming to us and they are talking about a variant and they are giving me information C.1,203 number C2T. And then what I ask them that, what's the transcript of your variant? They don't know. And we need to be extremely mindful that we all do not express the one isoform that you are thinking about. There are varieties of different transcript, and we need to know that exactly what transcript your variant is coming from, and then only translate that information to the right protein sequence, and then comes the right structure. So information needs to flow through all this information. Sorry, all these omics data type. And this is what it looks like. It's the real life example is you have a gene, and you actually have many different transcripts and isoform that can express in different cell types. And then comes protein isoform. And guess what? To make things more complicated, some people will refer isoform as a transcript isoform, and you as a protein biologist would assume it's a protein isoform. What is transcript isoform? So clarify, right, what we are talking about. Transcripts, and then to protein isoform, and then comes to the structure. And then if you are really working with experimental structure, they're like fragmented structures everywhere, right? It's hard to deal with them. That's why people like AlphaFold. So what we do is we need to take information from this transcript to the right RefSeq identifier, which is another database, because why not? We need to make biology complex, very complex. There are two transcript databases. One is called RefSeq, another is called Ensemble. Ensemble is maintained by Europe, and RefSeq is maintained by America. You can imagine now. I'll not go farther. Then we go to UniProt, and then we go to structure. So this is the connection that we wanted to do, and scaling this capability for the entire human proteome was the goal of Genomics to Proteins portal. So what we did here is we developed two modules. There were no two modules. There were only one module initially, which is the resource module, where we took the entire human proteome and we took variants from 3 different databases that you cannot see here. That is NOMAD, CleanVar, and HGMD. These are the 3 biggest genetic variant databases, one for population and 2 other for disease variants and clinical patients variants. And then we connected this data set through UniProt and AlphaFold and Protein Data Bank. And then we established this coordinates to coordinates connection. Now, when we established this connection, we realized that only seeing the mutation on protein structure is not helping. What we need is biologically interpretable features that are more known. So that's why we computed interpretable features or annotation for the entire human proteome, which looks like this, right? So for every protein, it's annotated with a lot of information and nothing is visible here. So I'm just going to outline what are those information. First is amino acid-wise physicochemical properties, structural features such as secondary structure, accessible surface area, post-translational modifications all through proteome, variant effect by MAVE, which is the saturation mutagenesis output collected through all over the world through an alliance effort. UniProt sequence features, so all the features that you can collect from UniProt, like domain information and DNA binding site, etc. Druggability features include pocket annotation, all the small molecule binding sites computed by 3 different softwares because one is never reliable, and then a whole suite of protein-protein interaction features. So that is how we annotate every residue of all the proteins within the human proteome. And this is a small overview for my 3 minutes break where I'm going to play a video. What this shows you is how to operate within the Genomics to Proteins portal, I hope, if it plays. Yeah, okay, it's working. So I'm going into the resource module. I just typed in MORG2. You can see some metadata information there, and I'm scrolling in. We have variant to sequence, variant to structure, and the general protein feature viewer here. So this whole suite of features or annotation I was just describing is present to the left. We have isoform mapping here. So see, if you don't pay attention, then you are missing regions in your isoform. And then we have the variant that is fetched from a transcript. There is no variant without a transcript information, literally. So we take variant information and map it to the sequence here. And the orange dots are the pathogenic variants from CleanVar. And one question you can ask is, why are they all on the N-terminal? Which when I see a protein and I see all the variants mapped, and this is a picture that we will often see, then comes the question, so maybe something interesting is going on. And which is interesting here is all the binding sites are here. These are ATP binding sites. It's a helicase protein, and there are ATP binding sites here. And then what we do is all the features that I was describing, you can click on an amino acid and you can see it all here. And the variant information card shows all the disease phenotype and the variant related data. The structural features. Okay. I guess I can talk faster than this video. Okay, so now we are going to variant to structure, and I clicked on an experimental structure, and now in this view, you have the sequence and structure side by side. So this is a homodimer of MORG2, and in a while, I'm going to click on a row, and that maps the data on the structure. And now I'm also mapping the missense mutation on the structure. And then you can, at the same time, map features onto the structure and analyze your variants in the context of the feature. So that's the purpose. And you can actually download this whole thing in as a PyMol session if you're more familiar with PyMol and want to analyze in PyMol. So the next section of the portal is the interactive mapping module. So we first developed the resource module, and then I started to have requests from the community that what if I do not have my variants public, and I have a de novo variants in a patient, or I'm generating synthetic variants that I want to analyze. So I want to be able to upload my own data. So one wise person once told me, do not pay attention to all the biologist requests. I ignored that at that point. And then we worked on developing this interactive mapping module where you can upload your customized annotation as variant score or features. You can upload your own structure because these days some people like ESM and some people like AlphaFold. So we upload both of them, and then we do this connection again. And after this connection, you can download the results, or you can even share the results with your collaborator if you want. So we share these results via a very secure email sharing. So you will add your collaborator's email address, and they will be able to log in using that email address and access the data that you have generated. Okay, so a couple of attributes and informatic features. So the scale, and I have demonstrated the user interactiveness. It was very important for that the biologists feel welcome and they are not overwhelmed with a GitHub repository. So see here, you can operate. It's not a GitHub repository. So it's a versatile platform, which was important because we can map mutations, scores, and features. And we made it as much as accurate as possible. Manual mutation mapping was a tedious task that I did myself for many years before I came up with this, because I was exhausted. And then we do the data sharing in a secure fashion. We have developed multiple APIs and Python libraries so that people who are more familiar with GitHub, who gets very impatient dealing with this, all this clicking and all these things, can access the data by your code. So it's very accessible, and the automation has been done in a way so that you get the updated data. Okay, so what are now different scalable applications? I'm just going to again swift through because I'll talk a few of them as in the method development section. So variant interpretation, it's a very demanding problem, and interpreting mutations using this context of interpretable features that I talked about is a scalable way to hint at the molecular effect. Why? Because you can collect a variant and can see this is their disease phenotype. And if it is listed like this in CleanVar, you know that they are pathogenic, but you don't know why. But when you see this in this context, when you see that this particular variant is on a disulfide bond, so it's going to break the protein in a certain way, either all misfolding or to certain extent. So that was one scalable application. Hotspot detection, right? Here, I know that we have a community of structural biology. It's not a new information that rare disease mutation clusters on protein structures. So identifying that hotspot is another scalable application. This is a new application and coming more and more. That is, we have a lot of chemical biologists around. They do regular base editor tiling mutagenesis, prime editing, and they need to interpret their data in the structure function context, which they are able to do via Genomics to Proteins portal. Fourth is the drug discovery, which is the genomic data. If you use them to prioritize your target, has a better success. This is actually an interesting information, and it was from a real drug discovery project that we were trying to select a pocket that we can target without hitting any other population variant. That's the goal. So for that, you need to map the population genetic data and identify pockets like this where the variants are all over other place and this place is constrained somehow. This is how evolution works, which is beautiful. So we identified that, and I think identifying pocket, keeping this information in mind is actually very powerful in identifying the right pocket. Okay, so I hope it was clear that the G2P was built to connect communities across. And we have a huge user base that has its pros and cons. I get a lot of email. Thankfully, there are people to address the emails. And if you want to know all about G2P portal, its YouTube tutorial is one of the most important resource. It has a huge set of use cases and small videos that you can look at and see how you can use them. And we always appreciate your feedback. And there is a feedback form right on the portal homepage. Okay, any quick question, or I'm just going to again swift through the method too. Yes.
So that's great. Thank you.
Yeah.
Super useful for hotspot identification, target topic, etc. Do you ever rerun the mutation back through AlphaFold to see how it?
Yeah. So in the G2P portal, you can do it PDB, AlphaFold 2, AlphaFold 3. Yeah, so it's literally one click. You go to your structure selection. So if you go to the YouTube tutorial, you will see you'll go to the interactive mapping, and there is in the video, I had really did it really too fast, but there is a list of experimental structure, and right below is the AlphaFold structure. AlphaFold 3 structure is only available through interactive mapping. We cannot make it publicly accessible without login because of the restrictions from DeepMind, because AlphaFold 3 is not freely available. That is not the structure of the mutant molecule. That is the structure. You can only see the location, because AlphaFold will not do any better in mutant structure prediction. So the structure you will see here are all wild type, unless there is a mutant structure present in PDB. That you will see here. But you will be able to see the wild type structure predicted by AlphaFold 2 and 3. Yeah. Okay. So the second method, I have actually talked about the background and the motivation for this. So I'm going to quickly skip through that and going to the method development. But the important thing is to know that we came up with this portal, and we now show users that, okay, if you have this mutation, these are the features that is going to perturb, and this will be a possible molecular effect. Geneticists and genomics folks came to me and said, we do not understand what you are talking about. I do not know what is a beta sheet. I don't even know what is accessible surface area. Give me something better. And what is better for them is a score. And then we can interpret them with features, right? That is why we developed these scores, which my postdoc who is developing it, she likes to call it protein feature enrichment score, or PFES. So we'll see. The background is very simple. With the Human Genome Project, we have an unprecedented amount of data, which is the genetic variants at million scale. And not all of them are pathogenic or benign. Do you know what's the most of the variants are? They are called variants of uncertain significance. That means we do not know how to interpret them. We do not have the right set of evidences to categorize them. And that is just the bulk of the data. 80% if you consider all variants, 85% if you consider missense variants. So that imposes a daunting challenge to the community that we need to be able to do something about these variants. And this amount of VUSs is growing every year. And it's going to forever grow. I never believe that we are going to solve problem. Can we help the community? That's the question. That's what we are trying to do here. So one way to help the community is to give them a score. And that is why there is a bloom of many, many variant effect predictors. Most of them, all of them actually, are machine learning based. As you all probably have heard about the tool called Alpha Missense. Very popular. Heidi Ram doesn't like it. I don't know if any one of you know Heidi Ram. She's the director of CleanVar and CleanGen. She likes to use Revel. But the point I want to make here is all these tools gives you a score, 0 to 1, pathogenic and benign. Then you have a threshold, right? I did something bad. Okay, it came back. Thank you. Okay, I'm not gonna move now a lot. So what I did is, I know that this is a very active field. So before coming here, I asked Gemini that how many distinct variant effect predictors do we have? And Gemini responded 118. Fantastic number. I was like, what? How, when and how I'm going to run my variants through all these variant effect predictor, and what I'm going to do, because they will definitely give conflicting interpretation. One will call benign, one will call pathogenic. So my point is the score enough? Is our score enough? As far as I'm concerned, it's not. So and now what I want to do is give a score, but also translate them and answer the question, why am I giving you the score? And the hypothesis is that protein sequence structure function information can help interpret missense mutations that are translatable. I might have my own bias because I love protein structure, and there could be other translatable way. And I always want to bring in, because variant effect prediction is a multidimensional task. Only protein will not give you all the answer, but then only evolution will also not give you the full answer. You need to try different ways. Okay, so we were very lucky that we had all the data in hand that we already gathered in G2P portal. We had the variant data and we had the protein feature data. With all that data, what we did, we ran a simple statistical test that which protein features are enriched for pathogenic variations compared to population variations, which gives results like this, that if it is greater than 1, sorry, I'm saying sorry to the cable. Yeah, I think it's coming. No. Okay. Okay. Yes, I'm seriously not moving. Anybody tell me, Sumaya, do not move if I'm even trying to move. Okay, so what it does is when it is greater than 1, then to interpret just one piece of result, the top one. So in pathogenic variants are twofold more prevalent in beta sheets of protein structure compared to the control variants. That is how to read these plots, right? Which we like to call, not I like to call, it is known. This is called a forest plot. I do not know why. Somebody needs to explain it to me. Why do we call this plot a forest plot? But that's what we call. Now, we can take all the 103 features that we have integrated in Genomics to Proteins portal and do the same analysis, right? And this is how these beautiful forest plots look like, which Silky in my lab has put together. She's a fabulous figure generator. I love her. Now, what we can do is, if you can see that all the features that are more prevalent in control variations are on the left side. All the features that are on the right side are prevalent in pathogenic mutation. We can now take any mutation in the world and identify their features, and we can sum up these scores and can have an overall score, right? That is basically what the PFES is. It identifies the features for a mutation and then takes all these odds ratios that I was just showing in the forest plot, and it sums it up. It's not a machine learning. It's not a fancy large language model. It's a very simple statistical method. But the good thing is that it's also very translatable. So now we can take this score of 7.52 and we can break it down into different groups of features that I was showing. That is this feature score attributed by physicochemical property or structure or interaction or post-translational modification. And from there starts our hypothesis generation, that what is the molecular effect and how to assay them. Okay, so now I have shown you this plot. Now this plot is taking all the human proteins together. We can repeat this whole computation by grouping the proteins into functional classes. And the results will be different because different components or motifs of proteins belonging to different functional class has different features that are associated with pathogenicity. For example, this top line is taking all the proteins together, and then the latter is showing you by taking all the proteins and grouping them into different functional class, 24 different functional class to be specific. What to look at in this very busy figure is you can see that the defense and immunity proteins has a way higher enrichment of pathogenic variants in beta sheet, which makes sense because they all form this beta jelly roll structure. And if you look at only the transcription factor, then the highest enrichment is in DNA binding domain, which also makes sense, which will not be the case if you take all the proteins together. So what we did is we repeated these whole computations by grouping the proteins into classes, and then finally developed our final score. I'm hoping that paper is going to come out very soon. So this is what you get when you have a gene and a target mutation. So you can get the full score and it broken down into different attributes to get an idea that where is it coming from. And then you can literally see and rationalize that score in the context of protein feature. So when you have an interaction component of 5-fold, then you can see that your variant is breaking a hydrogen bond in the protein data bank structure. So it's translatable, I hope. And I hope that a biologist will take that information and will design their assay accordingly. That's the hope. Okay, so we are using PFES to do a very large variant of uncertain significant recategorization. So can we use this score and information to recategorize them as high, mid, and low? This is a collaboration we are doing with Heidi Ram and Matt and Marina, who are from Broad Clinical Lab and also MGH. And we are also using this score to prioritize the rare disease genes and nominate yet not known genes where the variations would lead to protein mutations that we might never find out because those mutations are so lethal that we will never have a patient alive to sequence them and get those variations. Now, this is my favorite plot, which is a Manhattan plot, but not with GWAS result. It is with PFES result. And here, what we are demonstrating is all these genes that come up as a significant proteins where the mutation breaks the protein features in a way that they will cause a dysfunction. All the bold-faced gene names here are already known disease-associated genes. The ones that are not bold-faced are not known to be associated with any disease. Those are the new genes that we are prioritizing completely based on protein biology data. Okay, any question about PFES? This is my third method. Yes.
So when you compare it to the reference database, that's all things that are available in genome databases. I guess my question is, these are the reference sequences. Does it vary by ethnicity of the source?
Yes, they do. Yeah. Unfortunately, we cannot take that into consideration. I was actually talking to Amelia this morning that I do not even believe that the reference genome we consider as reference is reference for the entirety of the population. Yeah, that's a reference for European population mostly. Yeah. Not a good answer.
Of the variants that are for reference comparison set, there are ways to sort of classify those variants as severe or moderate using existing programs like polyclone. So maybe you could use a different relative to the two classes within
the population. Yeah, that is a great idea. Yeah. In the Genomics to Proteins portal, by the way, if you go, when you select, if you click on a variant and when the variant card information opens up, it will show you that which population the variant is coming from. That much only we can show at this point. But that is actually a great idea. That is basically calibrating your threshold based on the ethnicity of the people. Yeah.
test.
Right. Yeah. Awesome. Great point. Okay. So now my third topic here is so far we have talked about interpreting genetic variants. Now I'm going to talk about synthetic variants, right? So pretty much all the time in many chemical biologists and molecular biologists perform this CRISPR-Cas based base editor tiling mutagenesis data. And that data also needs to be interpreted in the structure function context. And what we have developed for that particular purpose will be the topic of this next 10 slides. Okay, so what is CRISPR-Cas based base editor? It's a technology to precisely introduce an edit in your genome sequence by delivering an editor, which can be either an ApoBec or a TAD-A, ApoBec doing A to G and TAD-A doing C to T, along with the Cas9, and then install that mutation, and then you can have a readout. And then the readout is mostly cell growth, which is not the best readout, but we have something that we can do high throughput. Now, it's extremely ideal for doing the residue essentiality and domain essentiality and structure function relationship assessment for not only one single gene. Very interestingly, it's suitable for a full complex, like you take 3 proteins that you know that bind to each other, and you can tile them all together. My largest study that I have experienced so far is 16 genes all together, like whole chromatin complex, repressor complex, we took and tiled them. So very high throughput and beautiful technology for structure function assessment. There are key challenges. One is that you cannot hit all the amino acid. There is a PAM restriction, sequence restriction. Not only you cannot hit all the amino acid, you cannot do all possible mutations either. So there is gap in both row and the columns. A lot of variables in the editing efficiency. In fact, the most challenging part is many chemical biologists do the assessment in mouse model, but eventually we are not drugging mouse, we are drugging human. So we need to do some translation from human to mouse and then derive the therapeutic hypothesis. No systematic hit prioritization. As I worked with more and more cancer biologists from Dana-Farber, I learned that all they do is they map their data into C-terminal, and then they decide on an arbitrary threshold of what makes sense. And now I do not know, but they feel really confident about it, but not very comfortable when I ask them that, what made you make that decision? And then they will say, because we just don't have a better way. Okay, I get it, but I think we can do something about it because data matters, and data gives us, right, the statistics, the right p-value threshold to choose our hits mindfully. Okay, so let's see how we did that. So we need a systematic method to expand the coverage, improve the signal-to-noise ratio, aggregate results that are performed across different environmental conditions and cell lines and species, and generate a consensus set of hits, right? That's very important. And then characterization of hits using interpretable features, which is linked to all I have been talking about like for last 40 minutes. Okay, so the method that we have developed is we will do a systematic quality assessment, taking the case and the positive control and negative control. Our positive control is always nonsense variance. Our negative control is always silent variance, and we'll see if they are very much well separated statistically. We'll take the base editor results, which are called log-fold change, which is the log-fold change of the guide RNAs present in a cell in between input and output conditions. This is what I'm going to use this word LFC so much in the next 5 slides. So this is what I mean when I say log-fold change or LFC. And then the third component is aggregation of the data. Okay, so we will go to the meat of it, which is this second module, which is B class 3D. So over and over, I faced something that is, as you can probably guess, that with TAD-A or CBE and APOBAC or ABE editor, we can only do A to G and C to T edits. What about the other edits? There are other edits that happen, right? Because they are not technically feasible, we miss a lot of other amino acid residues that are functionally close. Because we know how structural biology works, in structure, they are close. So what we have decided, what I once told to a chemical biologist, why am I not taking that score and assigning a relative score to the residues that are close to it, right? So that's basically the meat of this whole thing. So what we did is we took the LFC values, mapped onto protein structure. For every residues, we scanned a bubble, and we took the LFC values within that bubble, and we normalized the score. So that is a relative score. And then we have done the hit prioritization statistically, because by normalization, you will also add some noise, but we are only keeping the hits that has met our statistical criteria, which is p greater than 0.05. And because now I have normalized the score, and now we have more score, we also have the right statistical power to assign that p-value. Okay, so then the third component is the meta aggregation. So what we do here is, let's say you have done for the same gene, multiple screens, you sum up the values in every amino acid positions, and then you do again this hit prioritization, which is based on the stat, and you generate a consensus hit. And that's pretty much it. It looks very simple. It's not that simple when you have 70 screens per genes, which I'm going to show like in 2 slides. And then we have developed some result analysis method, which involves the hit clustering on protein structure. And we also developed the GitHub and Colab and the visualization, which we always do to cover both biologists and the folks who are more comfortable with GitHub not clicking through. Okay, so now what I'm going to show is what we can achieve by doing this LFC 3D score. So we took the published data by Brian Liao, which is an active collaborator of mine, varieties of different projects I have with Brian. So on the left, what you can see is the experimental result and the hits in blue. And on the right, you can see the LFC 3D score and the coverage. One thing is pretty clear, that the hit is almost 2-fold increased, but we are also covering the domains more comprehensively. And when I have done the amino acid analysis, then what we have observed that we can only hit certain amino acids by LFC, but with LFC 3D, we are able to cover a wide range of amino acids. Okay, Now I'm going to show you this result, which is from another of my active collaborator, Gabriel Griffin from Dana-Farber. And he did 67 screens for 16 genes in a chromatin repressor pathway, including a gene of my interest because of its association with neurodevelopmental disease, MORC2. And this is how the results look like, by the way, par screen. There are 67 base editor experiment that is performed here. And you can see the results grouped by human CBE editor, human ABE editor, mouse CBE, mouse ABE. What am I trying to show here, actually, by showing this? It doesn't tell anything, right? But what I'm trying to show is how there are editor-specific variabilities and gaps and species-specific variabilities and gaps, right? So we kind of aggregated them using B meta-class 3D, right? And this is what we see, these two lines on the negative side and the positive side after meta-aggregation, right? And then I mapped it using the Genomics to Proteins portal. And now what you see is that many sensitization hits are overlapping with the dimeric interface of MORC2 and the ATP binding sites. We are having sensitizing hits on the zinc finger. Those are still functional, but not yet any pathogenic variants. But that doesn't mean that they do not have an impact on the function. They are still functional variants. And then there are negative LFC hits that are almost covers pretty much the entirety of clean bar pathogenic variants, right? Okay. So the summary is it's a systematic workflow. Here I have put together a few collaborations that I have ongoing with Scott Armstrong, Srinivas, Matt, and then Gabriel Griffin and Brian Leah was there. And it's coming in G2P portal soon. It's not out there, but very soon. All right. Now this is the last topic I'll cover. Any question? I'm going very fast, I know, but I'm taking the questions through. Okay, if not, then this is going to be my fourth and the last method that we'll talk about, which is target discovery in the space of fusion-driven pediatric oncology. And the main reason for targeting this particular space is that every pediatric cancer is a rare disease by definition. So that is one of the target of the center. And also, I had a brother who had pediatric leukemia. He's living, but has not a very normal life. So now, background, right? What is a fusion protein? So fusion proteins are created by genomic relocalization of certain segments of two different chromosomes sometimes, and creating an entirely novel protein that we do not know anything about. Okay, disease focus is pediatric cancer, however, not limited. But what we need to analyze or do this method development is the dependency data. So I had this in my mind that I skipped that. How many here knows the cancer dependency data, cancer depth map? Anybody here is familiar with the data portal called depth map? Okay, so the depth map data does the CRISPR knockout and finds out that if you knock out one protein, what are the other proteins that are affected by it? So essentially what this portal records is that what are the two proteins that interact with each other for the function, because you knock one out, the other gets dropped out as well, right? So that data is important and absolutely necessary for running this method that I'm going to show. And the target mechanism is PPI inhibitor, and I'll demonstrate why. So this is how fusion protein works. So usually we have one anchor protein like MLL1, and then you have multiple different partners that it fuses with, and then it creates this novel protein, right? You want to target, if you want to target individual protein, that's going to be very costly. So we wanted to come up with a method to find a mechanism to hit all of them at the same time. Now, these fusion proteins often present either a druggable pocket or fusion-specific dependent protein that you can target. I hope it's coming clear now why we need that dependency data, because these fusion proteins are pretty much always undruggable. And I'll show you how, because these are novel protein and this is very spaghetti-like. I know with AlphaFold, we know what I mean by spaghetti, right? It's the all disordered region. This is an example. It's a very exciting example. So here on the left side, you can see a chromatin binding protein, MLL. And then on the right side, the transcription regulation, AFF1. And you can see the only domain structure we have in MLL is the set domain. All others are disordered region here also. When the fusion happens, then it becomes even worse because the fusion takes this part of the MLL and this part of the AFF1 and makes it an entire disordered protein. So we have a completely disordered protein that is not druggable. But do we have a dependent protein that is more druggable, which is the case here? Now, in this illustration, which is not made by mine, I forgot which link I collected it from, but it's a beautiful illustration to show that these fusion proteins do not operate on its own. It actually binds to menin to start the cell proliferation. So even if we cannot target this fusion protein, but we can inhibit this interaction, or we can target menin, we are good, right? So this particular method that we have developed is to ask this question and find the answer. Can we identify these menin-like novel targets and druggable interfaces in an unbiased and scalable fashion? So this whole workflow will start from the depth map data. So we did this for the KMT2A AFF1 with taking that fusion protein. We queried the depth map and we identified the cell line and all the other dependent proteins involved in it. And it nicely picked up menin, but it also picked up other proteins. That is what I want to make a point here, that there are other proteins it's dependent on. And then we did the structure modeling. And we developed a customized method to predict structure of the N-terminal and C-terminal of the fusion, and then connect them by biophysics mechanism, which is loop generation and energy minimization. That produced this structure, and now you can see how disordered that whole protein is. Only structured component is the menin, which is in the brown or yellow. We tried AlphaFold 3, it didn't work. Just to make sure that I show this, that it was all hallucination. It gave me a nice helical bundle, which is all false. Now, with that, what we can do is we can extract the druggable pockets, right? So I took all these 45 dependent proteins that I got from depth map, and I did this structured generation for each one of them. And then I extracted pocket using a tool that you might want to look at. It's a beautiful tool. The paper just came out from a Dana-Farber colleague, which is called AF2 bind. This has some mechanism to optimize finding their drug binding pockets, given it's an AlphaFold structure, which has its own limitations. Okay, and then what I did is we did the rank ordering, right, based on the druggability of that dependent proteins, and menin was the fourth one. So what it shows is we can identify menin as the druggable target for these undruggable fusions, and we can probably find more targets. Now, we need some experimental validation of this, that this interface is really the protein-protein interface. And there comes my beautiful collaborators, Gabe and Hans Pope, that you don't see here, but over there. As I speak, they are actually experimentally tiling all these 45 other dependent proteins and the fusion so that we can validate the binding interface. For menin, this is already proven because menin had already existing base editor data. So we took that data and we ran the B class 3D method that I demonstrated using the structure that we predicted using AlphaFold, customized AlphaFold 2 method. And we identified not only the recurrent mutation site, but the entire interface where the drug binds to. Now, this is an FDA-approved drug for menin. So next question is the last question here in this workflow is, we were lucky that here we had an FDA-approved drug. What if we did not have an FDA-approved drug? Can we run a virtual screening? I do not know if you know this. It's called molecular docking. So we can take a library and can dock it in the structure, and we can nominate hits. So that's what we did using a broad library plus the 33 known menin inhibitor. And we were able to identify revumenib with a binding affinity of -9.7, which was very cool. And which leads me to the summary that MEN1 can be identified as a drug target and the revumenib can be identified as a hit in a very unbiased fashion, even if we didn't know anything about it. Okay, so we are almost done. I hope I was able to demonstrate that although these methods that I have just showed they are very diverse for different purposes, we have two themes in common, which is we are working across different omics field, protein-based editor data, depth map data, structural biology, and all this to ultimately inform drug discovery. And the second thing is collaboration, is catalyst. We start with sequencing or experimental data, then we go to machine learning method, we develop method, we generate hypotheses, we validate them, and we loop through, right? That is very important. Looking ahead, I always like to end my talk with what I am excited about in the next couple of years. I think in the next couple of years, what is coming more and more is the gene editing as therapy, which I'm extremely excited about. I think we have come a long way from 1987, which is when I was born. And the term genomics was coined. By the way, I didn't know it. Very recently, actually, Eric Green, who came to the Broad to give this talk, and he was saying, do you know when the term genomics was coined? It was 1987. I was like, oh, look at that. Some good things happened together, right? Okay, anyway, sorry, that was narcissistic. Anyway, so from Genomics to Human Genome Project to genomic medicine, and now we have baby KJ, who was treated with gene therapy, right? And I really believe and want to believe that there will be more cases of such, where the methods of such that I have demonstrated so far will be utilized and used to identify more of these cases. Second thing that I'm very excited about is the tango between human and AI, right? And here the tango equals to collaboration. As I speak here, some fantastic software developers and machine learning engineers in my lab are developing AI agents in each of these data space and connecting them through, so that here we do not have only humans working with the data in the G2P portal, but we also have agents that will do tango with us, because just the way it takes two to tango, I really believe that almost in every field of science or economics or finances, we will need human and AI to tango together, which is human and AI collaboration. We'll see how far we go in that direction. Okay, so with that, this is my last slide. Thank you all for listening. Thanks to all my collaborators and my funders. I'm hiring 2 data science interns in the next couple of months, if any of you are interested. And with that, that's it. Thank you very much.
You can talk with the questions for her. So many great tools.
Yes.
Part of trying to be short-winded, but it made me think of it when you brought up the druggable targets, like on your research. And I know we had a good discussion earlier about dynamics, so ignoring that. Some proteins, like I think it's like the ABL1 kinase, which causes phylocythemia through chromosomal translocation.
Is the ABL we are talking about?
Yes.
BCR ABL fusion? Yes. I actually have 2 slides I didn't show. Yes, go ahead.
It occupies 2 stable conformations, DFG in and DFG out.
Yeah.
Stable enough that experimental structural biologists are able to get X-ray structure and cryo-EM structures. So it's uploaded in the PDB as kind of 2 separate entries. So when you're aggregating the data to develop these methods, how do you account for multiple entries with multiple conformations of the same protein? And kind of the second part to that question is, if you do you weight it depending on the amount of time that a given conformation is spent in? Like the ABL1 kinase, it spends 95% of its time in the DFG in conformation and only 5% out, and that is really, really important for like druggable specificity. So if you take that into account, aggregate it.
So first of all, to identify ABL itself as a drug target, we do not need molecular dynamic simulation, right? What we need the BCR and we need the ABL, and we find the dependent protein, we dog them, and we predict our own structure, and hopefully a similar to PDB structure will show up. And by comparing them with all that are available in PDB, we need to identify this is open and this is closed, and which one we want to target, right? For BCR ABL, what happens is you lose the regulatory region called CAP that keeps the kinase always on. So we want to turn it off, right? So we clearly know that we want to inhibit that conformation. And we do the virtual screening or molecular docking on that conformation. Does that answer your question?
Yes, you're using like a consensus structure?
Yes, we consensus and also a little bit expert driven, right? So you need to talk to structural biologists. You need to show them that, and you need to gather opinion and then select it. Sometimes you just need to go empirical, right? Do it both ways and see what works. But then what I prefer is talking to structural biologists. Every time you have to make a decision, you need to find out who else I can talk to, who knows better than me, who can guide me better.
Yeah, there's
Looking under the hood and into the black box.
Yes, XAI, yeah. Yeah, so what we have done, and we actually have a bioarchive paper about that. So the idea of this is we have this a plethora of different LLM going on, right? I can name like on the top of my head, ProtT, PROST5, ANC, ESM, AlphaFold 2. And what I want to do is, let's say I'm doing a prediction, I'm developing a prediction model. So I want to take all the ion channel variants, which is an active project. I didn't show that. And I want to identify which variant will lead to loss of channel function and which one will do gain of channel function, right? And then I want to use the LLM embeddings and do transfer learning. So layer it up with another multi-layer perceptron or random forest, and then to be able to do that prediction, right? But the question is which model to choose, right? So we want to answer that question by taking the embeddings of those models and do not run machine learning model for every LLMs, but take those embeddings and try to see which embeddings correlate with known biological information. So for example, we do know that SCN2A has a fraction of variance that causes autism, therefore loss of function, and some fraction that causes epilepsy, therefore gain of function. So which LLM in their embedding space captures that information more in terms of statistics, correlation, and association, and then we choose that model and do the training, right? And this tool actually has a Python library. It's really by statistics and visualization. It's generating that prior information to make a mindful choice. Does that answer your question? Yeah, okay. And that's a tool called MIEMB. We are actually very close to the publication, second round of revision.
well, thank you so much. There's no more questions.
Thank you. Thank you. Thanks, all of you. Yeah, that was very fast.
No,
Thank you.
fair. Yeah, I noticed that when how many regulators you found. I was like really excited.
I'm sorry?
We actually work on the fly version of MLL in
Yeah.
my lab, so I was like, this is so cool.
That's
I
my
Yeah.
I all of a sudden.
Yeah, I mean, this is the base editor tiling screening. I'm just so excited for that data that will come up.
Amazing to see
Yeah,
how that
right,
all looks.
Right. And isn't it great? Like, I marvel at the capacity of the technology. Like, how do you tile all 46 genes at the same
That's
time?
amazing. Yeah, thank you.
Thank you.
Wait, did you want to meet back here for dinner?
Yeah, we will have an individual meeting. I will have you several questions.
Oh, sure. Look, so Yanma, so she, hey, yeah, nice to see you again.
Yeah. I wanted to let you know, I'm not doing grad school next year, but I'm gonna be working at the Broad in Evan McCaffrey's lab.
Hey,
Yeah.
so good.
Yeah.
When are you starting?
I start in mid-August.
Beautiful.
Yeah.
Stop by. I'm at 75 Ames, 5019.
Okay.
And we are organizing, I'm chairing the Machine Learning for Drug Discovery Symposium again.
Awesome.
Yeah, so we'll meet.
Yeah.
Oh,
Yeah, no, I'm super excited though.
it's beautiful.
Let's
I'm
see.
glad you made your decision.
Yeah.
So I was thinking about you, that
No,
he has to
yeah,
make a decision.
Yeah.
Yeah.
So I'll be there for like at least a few years. and-
Beautiful.
Probably
Yeah,
PhD.
come to our space. It's on the 75 Ames, 5th floor.
Okay, all right,
Yeah.
okay.
Yeah.
Awesome, yeah. One second.
care.
Good to see you.
Same here. Hey, are we staying here?
Yeah, do you need a break?
No. I just need to drink some water. And I was so conscious about
by yourself or?
from here. I was always so conscious about this thing because I feel like it was heavy. It's like
Yeah.